Well, hey AP, hope you're doing well when you watch this video, and again, super proud of you for how hard you're working, and we're on this journey together. So, thank you so much for taking your time to watch this video. I hope to be helpful. I know we've gone through all this in class, but it's always helpful to review again. And if you have any questions, maybe I can answer by using this video. And so let's just talk about cell surfaces, modifications, and also cell signaling in this video. Okay. So cell surfaces in animals, the extracellular matrix, of course, stuff around the cells. There's lots of different proteins in here. I probably wouldn't necessarily memorize these per se, but they do have a function in like resist stretching and resilience, signaling, and also regulating passage of material. All those proteins do have a play, and here's kind of what it looks like for as far as a animation, a scientific drawing. It can vary in quantity. Bone, cartilage, epithelial tissue all have different types of the extracellular matrix. There's junction between cells. I would know these, important to know just in general. Adhesion junctions attaches to cells. Desmosomes, I know I brought it up in class about how some cardiac arrhythmias are due to desmosomes not working correctly. Tight junctions, of course, impermeable barriers. So this is like your skin. Your skin's a great watertight junction. And then gap junctions allows communication. Why? Because ions and various molecules can travel through them. And so plants do need to be able to communicate with one another. Plants have a freely permeable cell wall, which means plant cells can communicate to one another. Of course, a plant has to maintain homeostasis, and communication is a big deal with that. And so signal transduction. Cells they don't have eyes, noses, ears. So how do they respond? How do they respond to the world around it? Here's some objectives. Definitely, if you know all these, you should be prepared for the quiz. So in multicellular organisms, cell-to-cell communication allows the cells of the body to coordinate their activities. Again, this is multicellular organisms. And so, like us. And so communications is essential. I mean, we're maintaining homeostasis, and it's essential for a lot of different functions that we control, okay? Even when you're feeling stressed, that's communication. And of course, y'all can remember from a lot of our class about all the different things that we went through. But you can see here that it's similar to, electrical impulses are converted to sound waves in like old rotary phones. And so an extracellular signal is the input, and then the output is of some sort of response, okay? And so cell signaling, the chemicals are called ligands, okay? It could be physical stimulus like heat or light, or touch, following receptors, activation, a signal transduction pathway. I would definitely know all about signal transduction pathways, okay? Very important. And so different signals, these are definitely important to know and memorize. Autocrine is the same cell, paracrine is nearby cells. Juxtacrine requires direct contact, and then hormones are part of the endocrine system in humans, and so it travels to distant cells. Okay, so very important that you know those four. I would definitely make note cards about that. There's another more specialized type of local signaling, and this is the synaptic signaling, or the synapse. And so we talked about that when we had our marijuana debate, and I showed you that that Flash website, the website that has the Flash movies on it about the different drugs like the benzodiazepines, caffeine, alcohol, nicotine. We went through all of that, and that's the synapse, right? And so how you feel is basically how your brain is communicating and what's molecules around those synapses. And so here's just a picture of showing you all that. We went through glucose, the example here, and that glucose is can be seen, right? When glucose is high in the body, insulin is released. So that is a response to a signal like glucose being around. But glucose has to be inputted into cells, like brought into cells. And so that's not good. And how does that happen? Well, insulin has to bind to a receptor. That creates a signal transduction pathway inside of the cell, which leads to this protein being made. Okay, and so it's kind of seen like this, right? So here is the signal or the ligand. Here's your receptor protein. Here's the signal, all of this right here is the transduction part, like the signal transduction, all the responses. And then the response is right here. This is the response. We're making this protein, and of course, proteins have phospholipids, therefore it fuses well with the plasma membrane, and it lets glucose come into the cell now. without this protein right here, glucose cannot get into the cell. And this protein is the response to this signal right here. One bad apple spoils a bunch, right? And so this is ethylene. Ethylene is a gas that is also a signal that increases the ripening of fruit. You can see right here, this is kind of the pathway, but once again, signal, binding, response, okay? Only cells with the necessary receptors, of course, and so it involves a signal, a receptor, and a response. Very important. Know all about signal transduction pathways, okay? And so the reception is the detection of the signal. Again, you can't receive the signal unless you have the right protein. Transduction is the conversion of the signal to a cellular response via a pathway, and then we're trying to get a certain response, right? I went through this, and I told you to star this when we talked about it, and I said, listen, if you can explain this out loud, you know what you're talking about. 3 parts, reception, transduction, response, super important. That's just the basics, okay? And here's just another example of that. So signal transduction pathways often include allosteric regulation. It's never going away. Shape change, never going away. So proteins change shape as a result of a molecule binding, okay? And this might create that protein to be active or non-active. And it could create the active site to actually be available. As a review, allosteric regulation is on a site other than the active site. Okay, so here's a regulatory molecule. Here's a regulatory molecule. Some regulatory molecules turn the protein active or on. Some of them turn them off. Okay, there's different types of receptors. They can be inside the cell or on the cell surface. And of course, in intercellular receptors, we would expect the ligands to be small and nonpolar. That was on the test we just took, like estrogen, hormone. On the cell surface, must be large or polar. They cannot diffuse across the membrane. We would expect that. Example of that is insulin, peptide hormone. And so reversible binding is important because cells need to, listen, you're maintaining homeostasis. We need to stop responding. We're not going to need to respond to a signal indefinitely. We have to turn the signal off. And so in order for that to be the case, we have to have the appropriate response. Inhibitors or antagonists can bind in place of it. So for example, the most widely used drug on the planet, caffeine. It binds to receptors, blocking a normal ligand, which is adenosine. And so adenosine makes you sleepy. And so caffeine is an antagonist. It binds to the same place that the ligand does. 3 different types of receptors: ions, channels, protein kinase, and G-protein linked receptors. Ion channels, kind of what they look like. Ligand gated, which means the ligand binds, the channel opens, and ions rush in. We need this to be able to use our muscles correctly. You can see right here, conformational change, it's open, ions rush in, very important. Protein kinase. A kinase is a protein that adds a phosphate. Have to know what a kinase is. And so not all protein kinases are receptors. There's them that are just enzymes. But here's, again, kinases. Look, kinases add a protein. And then phosphatase is the opposite, and it does what? It removes the protein. Okay? And you can see right here, here's just another picture of all of that. You can see the signaling molecule, activated relay molecule, and then we have an active protein kinase. And now we have another active protein kinase. And now we have another active protein. And all these proteins are activated because they have a phosphate group attached, thereby changing the shape of it into the conformational shape change, and now it is active. And now you have the cellular response, which is good. Ligands bind to G-protein linked receptors, expose a site that can bind to a membrane protein, a G-protein. The G-protein is partially inserted into the lipid bilayer and partially exposed. So let me show you a picture here. So here's an example of the G-protein, partially embedded, partially exposed. Signal activates the G-protein. GTP is just a type of energy. Yes, there are different types of energy than ATP on a cellular level. And then you have an effector protein creating products and cellular responses. Okay? So here's your G-protein coupled receptor. Here's your signaling molecule. Activates it. Now the G-protein is activated. It activates an enzyme, and thereby you have this whole cellular response. I'm not going to read through this. Of course, GPCRs are involved in cancer initiation and progression. We went over this in class. How important is this? 800 GPCRs examined to date make up the largest family of cell surface receptors in mammals. G-protein receptors, okay? Intracellular receptors are found in the cytoplasm or nucleus. Again, what type of molecule will go into it? Smaller hydrophobic. So hydrophobic are the steroid, thyroid hormones, nitric oxide. All of those will go directly through like this. Aldosterone is a steroid hormone. You see right here, it diffuses right through, non-polar. Aldosterone binds to a protein, activating it. The hormone receptor complex enters the nucleus, binds to specific genes. So we're looking at a specific chapter, reads those genes, which causes that gene to be expressed. Now we're making a new protein, and that is the response. Oh man, I shouldn't have started writing with a mouse, but I'm going for it. That's not very good. Okay, response. And then, of course, this leads us to the whole $2 billion testosterone industry. It's probably even more now. And so here's some questions we went over in class. Be able to answer those is helpful. And so signal activation of a specific receptor leads to a cellular response, which is mediated by a signal transduction pathway. So signaling can initiate a cascade. So it's like this, right? You flip over one domino, and then all the other dominoes fall down, right? And so one domino, or like the Rube Goldberg, video that I showed you in class, the machines. It's a cascade of protein interactions, and then it can be amplified and distributed to different to different causes and different responses. Okay, so cells change in response to signals. This is the environment. Okay, opening of ion channels, upregulation, downregulation of genes. Genes are kind of like chapters in a really big book. We don't need to be reading and making everything every chapter says all the time. We would lose energy, homeostasis, all of that. So how do we know when genes are being expressed, which means their products are being made? Well, that is cell signaling. Alteration of enzyme activity is another example of that. Okay? And so quorum sensing. This is a great video that I would watch. I would watch this video if it's about quorum sensing. And if you just look up quorum sensing TED Talk, it's great. But basically, this is an example of how do bacteria know when to start doing different things. So it's all about molecules on the outside. So that when the extracellular concentration is less than intracellular, cells continue to replicate. However, this molecule keeps being made just by biological process, and eventually, extracellular concentration, the cells stop replicating, and they start doing different things and responding as like a unit, which is a pretty crazy thing. But I'll watch the video. It is pretty helpful right here. EpiPens, we're all kind of familiar with those. Epinephrine, system-wide effects, right? And what epinephrine does in one part of the body does something completely different in the other part of the body. So it opens lungs and increases blood pressure. It decreases swelling and reverses anaphylactic shock. So opens up the lungs, but it constricts blood vessels, which means raises the blood pressure. But it relaxes, airways. So it relaxes one type, constricts another. And so this is an example of how the same signal can lead to different responses in different types of cells. The signal transduction pathways, why? Why do we get different responses? Because the pathways are different in the two cell types. Okay? So the extracellular signal molecule, the ligand, that binds to the receptor is a pathway's first messenger. Secondary messengers, so first messenger. Now we're going to dive into secondary messengers in CAMP. Okay. Second messengers are small, non-protein, water-soluble molecules spread throughout the cell. Cyclic AMP means 1-phosphate, and calcium ions are examples of secondary messenger. CAMP, so people like to write this as CAMP. CAMP is a very important molecule for us. Like that. It's written like that. CAMP. Need to know about CAMP. Okay? And so is widely used. So adenyl cyclase, an enzyme in the plasma membrane, rapidly converts ATP. Oh man, no, my bad. Rapidly converts ATP to CAMP. CAMP is usually the activation of what? So this is all about reception, transduction, response, the three parts of a pathway. And so this is all about transduction. We are amplifying a signal. We are causing a signaling cascade. And so the immediate effect of CAMP is usually the activation of protein kinase A, which phosphorylates, which means adding a phosphate group to a variety of other proteins, okay? And so here is a G protein receptor, but this is also introducing the concept of CAMP. And so you have a first messenger, okay? First messenger signaling molecule such as epinephrine. Okay, so here's epinephrine. Here's a GPCR. Binds to it, conformational shape change right here, okay? Shape change. G protein's activated. Activates adenyl cyclase. Again, just knowing the general flow of this would be helpful. Activates a lot of different other stuff via a secondary messenger, CAMP. What does CAMP do? CAMP activates the other proteins, thereby increasing cellular responses. So first major molecule is epinephrine, but the second major molecule is CAMP. So secondary messengers regulate target enzymes by binding to them non-covalently, which means we can turn the signal off. They allow the cell to respond to a signal membrane event with many events inside the cell. They distribute the signal. So they allow the cell to respond to a single membrane event with many events. That is called amplification, right? You have one signal and then you amplify it into hundreds and hundreds and hundreds of other molecules being made. That's called amplification, okay? This is what CAMP looks like. Don't need to know that, but you can see just one phosphate group. There it is, bam. All right, so here's a great picture to kind of review a little bit. We only got a few more slides left. Thank you for your attention with all this. So you can see epinephrine right way up here, okay? Receptor, G protein, adenyl cyclase, good old CAMP, right? But watch amplification. We go from one ligand, epinephrine, to 10,000 glycogens being converted into glucose. So when epinephrine, let's say you're afraid or scared, epinephrine is involved with the fight or flight response, right? You need rapid energy because you're about to flee or fight for your life. And so that rapid energy comes from epinephrine binding. And so it activates protein kinase. Okay, that protein kinase inactivates glycogen synthesis. Glycogen is made from excess glucose. We don't need glycogen being made. We need glucose right now, stat. So we need to inactivate glycogen synthase. It activates another protein kinase. It activates this protein called glycogen phosphorylase, which phosphorylates glycogen, releasing stored glucose molecules from glycogen, okay? And then thereby we have our glucose, and that's our response. We need that energy to fight or flee, okay? So 20 molecules of CAMP are made. The protein, let's read right here. The protein kinase cascade amplifies the signal. Here, for every molecule of epinephrine bound, 20 molecules of CAMP are made, each of which activates a molecule of protein kinase A, okay? And then that leads to 100 activated phosphorylase kinases. That activates 1,000 and then 10,000. So this is called amplification. So the original signal is amplified at every step, every single step, okay? Signal transduction ends after the cell responds. So enzymes convert each transducer back to its inactive precursor. We have to be able to regulate signals. We have to be able to turn them on and turn them off. The balance between the regulating enzymes and the signal enzymes determines the cell's ultimate response. So here you go, right? We talked about it before. Kinase adds a phosphate. Phosphatase removes a phosphate. Inactive and active is all based upon what? Come on now. You should know where I'm going right here. This is all about shape determines function. This is all about that. That's what this is about, okay? Then you have your receptor, an active G protein, activated because it has a GTP, GTPase removes the phosphate. Again, here's adenyl cyclase, right? Adenyl cyclase, this is activated. CAMP, oh my bad. CAMP is activated, right? Phosphodiesterase is an enzyme. I would actually know that enzyme. That's an important enzyme to just kind of know. Super important. Inactivates CAMP to just AMP, which can be funneled back into the ATP kind of pathways. And so important to be able to turn off signaling when we don't need the signal anymore. Fight or flight's great, But if you can't get out of fight or flight when there's no danger around, that's what leads to panic disorders and loss of homeostasis and problems, right? Cells can alter the balance of enzymes in two ways. This is a really important slide as we end right here. Enzymes, right? We've been talking about kinases and phosphatases and phosphodiesterases and all these different enzymes. Cells can alter the balance of enzymes in two ways. They can make it more often, break it down more often or not, but they can also let's say it's made, right? So we can make it or break it down. But let's say it's already made in activation. Well, it can be in an active form or an inactive form. Okay? So two ways. We can either make it more often or break it down. Okay? Or if it is made, we can activate it or deactivate it. This is all about shape determines function. Wow, don't know how to spell. Okay. And so, how do I erase this? I guess I can't. Okay, well, there we go. Maybe not. Caffeine blocks. So if caffeine blocks the enzyme CAMP, phosphodiesterase, can you explain how caffeine augments the fight or flight response? Augments means makes it stronger. Well, I'm going to let you pause it and think about it. So pause. And let's go back up here. Phosphodiesterase is right here. So if CAMP can't be broken down anymore, right? Phosphodiesterase breaks down CAMP. If it can't be broken down anymore, it is still sending that signal. CAMP is still sending the signal to release more glucose because we're in danger. We need glucose now to fight or flee. So caffeine, yes, it augments. It makes the fight or flight response greater. And then you can kind of read an article about caffeine. I hope this was helpful. I still don't know how to get this off the screen. Oh well. I hope this was helpful. I hope please reach out if you have any questions. God bless, guys.